Good evening, participants. Um, shall we start the session? Good evening, all. Sorry for the delay. There was a slight network issue from our side. Any doubts in the last session, participants? So if you have any doubts or queries, we can clarify so that it would be, I can move to the next session. So have you worked out with the databases? Have you checked the PPT and worked out with the databases which taught in the last class? Participants, if there is no query, just give a thumbs up symbol. So I'll be going to the session. Am I audible? Just anyone? Okay, I got some reply. Thank you so much. Okay, I'll share my screen. So today we are going to see about the compound-based data collection. Compound-based data collection, that is the compound target interaction database. As I told earlier, we are going to see in depth about the PharmMapper binding database and Swiss target prediction database. So we are going to see about these three databases. And so coming to the compound target interaction database. So as the name implies, based upon the compound structure features, or based upon the compound inhibitory nature, based upon the compound pharmacophoric features. So based on any of these activities, the compound data will help us to predict some targets. That is the protein, enzyme, receptors, and all those things will be predicted. So that compound might be, as I told earlier, it could be a natural metabolite or a synthetic molecule. So based upon its features, structure features, the targets would be identified from the databases that I have mentioned. So these databases will serve, so actually this databases will serve as a knowledge base in the drug discovery to identify and to analyze the pharmacological, toxicological, and systems biology data and help us to understand more detail about the molecule inside the living system. So our ultimate aim is to understand the mechanism of the molecule with the protein and mode of action. So for that, this particular, based upon the features, when we take a target, it would be better helpful. So so as we are talking about compound-based target database, so there would be some features generally we usually require or types of data that would have been stored. Usually the compound structure and property data. So mostly the structure input, your input structure would be the SMILES format, and based upon its structural properties, so based upon the structural property, the data structure, target structure would be retrieved. And biological target data, based upon the protein name or uniprot pathway diseases, we analyze the data. So as we know, once we get the biological target data, that is about protein data, we go for string analysis to identify the pathway and the disease mechanism relevant to this. So interaction-based data, for example, in case of binding database, the mostly it would be like constants like KD, KI constants, inhibitory constants. So an IC50 value and EC50 value, that is inhibitory concentration value, and all these value help us to find out the compound with much binding affinity towards the target, which was an assay-based result. So more approved target, we can say in that case. So we can take that target also for our reference, for our studies. And clinical or therapeutic data, that is approved indication and clinical trial status and adverse effects of the drugs. So based on that, also we'll take the target data and predicted data, that is the prediction based upon some of the features for which the experiment data won't be available. For example, farm mapper, all these in these databases, it is purely based upon the prediction mechanisms. So hope I'm clear till now. Any queries? Hope there is no query. So let me go into database. The first database, which is a binding database, which contains the experimental protein or a ligand binding data. So I'm just going to tell you about how to search this particular database and what were the output we get and what were the key parameters we need to check over in this particular database. And Farm Mapper, it is based upon its pharmacophoric features, that is donor, acceptor, aromatic ring. So these kind of things help us in identifying the target, and how we have some scores and what is that scores and how to retrieve the data based upon the score. And Swiss target prediction, it is the ligand based target prediction tool where we submit the SMILES format of the compound, and we get the list of target, and based upon the score, there is also a score called normalized score with which we interpret the results. So first database we are going to see is about binding database, where this experimental binding data that covers a small molecule interaction. It was purely based upon the dissociation constant, that is Kd value, inhibition constant, that is Ki value, and half maximal inhibitory IC50 value. So mostly in literature we would have found, and this database was created from the literature evidence of the target and the compound data, which was primarily available. And this primary target might be enzymes, GPCR, ion channel protein, or kinases, which might be linked to the human disease. So purely this binding database is a molecules for the human-based diseases alone. And next, coming to the ligands, small molecule that has been tested experimentally. Those data alone will be available. Not the entire set of data won't be available in case of binding database. So for the molecule for which the experiments or already the wet lab experiments were carried out for that kind of data. So here that was very minimal target we get in the binding database when compared to other database, as it contains more of experimental data, but the data which is very much important. So what were all the different column of data I get in the binding database? So first I get about the target name, that is the protein name, kinase or receptor, whatever it is. Here we won't get the gene name. So once we get the data from this particular database, we need to go to UniProt database to get the gene ID for the particular protein name. And next is the indexes of the uploaded compound yielding hits. The indices or the IDs are nothing but the match target pharmacophore that helps us in get the information about the hits which the targets are. And the similarity, maximum number of similarity, the score would be 1. It was very good. If it is, usually we have a threshold of 0.7 so that we get not least match, or we get the best match, that is below 0.7. It would be like so below 0.7 there would be like it would be a very lesser match, and if it is after 0.7 it would be better match, and you get the interaction, that is it's for the all compounds. And so the data set, total number of compound data set you get for the hit produced, and links to the external database would be there, and also a workspace and the collection for the further analysis. And last one would be the download option. So these are all the parameters we are going to analyze in the binding database. So till now, the theoretical part of binding databases I have told. Any doubts, any queries, participants? If there is no queries, just give a thumbs up symbol. We'll go to the next slide. Any queries, participants? Any queries? I'm just checking. No queries, just give a thumbs up symbol. Okay, fine. Let me go with this homepage of binding database. Here, this is the homepage of binding database. You can see the information, that is compound, publication, specialized database. And here in the left inside, you have a specialized tool icon over here. You can see here in the left inside, uh, in that you have find my compound targets. So this data will help you to fetch the compound based targets. And once I I paste the compound, I get the list of target. That is the page where I'll paste my compound in smiles format, and I give the search option and the parameters I have to adjust. I'll get the list of targets. So I'll just show you practically how to paste and how to retrieve the target. And list of targets would be listed, and you have the values, that is. As I told you, this value should be 0.7 above. So for my compound, I got around 40 to 50 targets. So I have one over here at the top. Later, it would be around 0.7, 0.6 like that. I mean, that is hits over here. And the maximum similarity value, I have kept the threshold 0.6. So after that, the compound were listed. And you have the IC50 values that was mentioned and links to that. And lastly, you have two-dimensional and three-dimensional data to get retrieved. So this is how your target names should be. Once you get this data, you have to you have to copy this data into an Excel file, and you have to find out the gene name using the UniProt database. Am I clear till now? So this is a step to retrieve the data from binding database. Any queries in this? I just show you practically. Any queries, participants? Okay, I'll just repeat once again. Thank you so much. Sure. So this database contained the find my compound target link were there. So once you click on to this particular link, the page will be open. There you have to either paste the smiles format, or you have to upload your SDF file format. And you have to adjust the threshold value to 0.7 that I show you practically, after which you get the page like this. That is, once your compound have the experimental data or the target for it, you get the data in this format. So it will list out all the data. Here we cannot download, as that even though download option was not available, we cannot download it directly. You can copy the entire page and paste it in Excel and have this target names alone. With this target name, I have to take the target name, go and put it in UniProt. I'll just show you practically when we do. When we put it in UniProt, we get the gene name for UniProt. For each target, we get the gene name. So the copy pasting work is there, because we may get 50 targets or 20 targets depending upon our compound experimental data availability. So for that, we get the data and we have to copy paste and work with the particular data. Is it okay? Nandini, is it okay? Any other queries, participants, till now? Fine, I got it. So let me finish this, or shall I complete all the three database and go to the hands-on training part? Shall I complete theory part first, then go to hands-on part? Just any one response? Okay, fine, sure. Thank you so much. So next database is PharmMapper database, where the target prediction is purely based upon its compound pharmacophoric features. That is the hydrogen bond donor, acceptor, and aromatic rings, basic features of the compound pharmacophoric nature, and used to 3D query of your molecule and compare it against a large database, that is the pharmacophore model, derived from the known protein targets. Already for the protein, compound-based target with the protein matches, the databases will have the data. So that data, based upon the compound structure you are giving as input, it try to fetch out all the target, and it fit with the ligand based upon the target based upon its pharmacophoric features, and we get around 300 target maximum, and minimum based upon the compound features, it might be 100, 150, depending upon our compound feature. And also we have the scores to segregate in case we have more number of target. So that also I'll just tell you. But this database is ultimately to find out the compound data. Before I submit the data, I have certain option, that advanced parameter option. So where I can adjust the parameter or population size, energy minimization parameter, force field parameter. So all these parameters, if there is any change I require, I can actually adjust this. So this 𝐏𝐥𝐞𝐚𝐬𝐞 indicates that indicates the genetic algorithm which was optimizes simultaneously. It includes the pharmacophoric fit, and this value, if it should be 4 above, means it would be good. Default parameter was 4, and number of genetic run 1. So if you want more of number of runs, and if you want to ensure your results to be more iterative, so as we do in Autodoc, usually to perform many iteration, we usually increase the parameter. So if you want to change any parameters, you can change, but based on the server, because the server usually very slow server, and sometimes it won't work. Definitely the server has certain issues also, but still, if it works, it will give you the list of data sets and that data set we are going to take it for the analyze. I'll just show you how to submit the data set. So this is the homepage of the PharmMapper, where you can see the submit job option. So once I give the submit job option only, I'll get this particular page where I have to upload my SDF format data. So we know so I have already told about the how to download the SDF structure from the PubChem database. So that structure you need to download. Suppose if I'm going to work with the quercetin, so I have to download the quercetin data and SDF file, and I have to upload the browse and upload over here. After which I have to submit my mail ID and quercetin compound name. That is job description I have to use. Whatever the compound name I'm submitting, I have to use that. So these 3 data is required to submit your data. After which I have told you the parameters. Maximum generator confirmation, it should be 300. Or so if you want 100 top hits, you can give 100, or if maximum number can be 300, so that also you can select. And objectives used in Mocha, so the numbers, if you want, you can adjust. Otherwise, if you don't know about this, so if you enable this option only, it will show, otherwise it won't show. And you can choose the force field. Energy minimization, yes, if you give and choose the force field, then it will take more time to optimize your target. So you can try to use this option, but generally go with the default option when you do it first time. And here you have 4 set of option. So here I'm just selecting human target only. But in case if I'm going to work with microorganism based disease, SARS-CoV, something like that. So in that case, if you give all targets only, it will give you the microorganism based targets also. In case if I select human targets only, it will list out only the human-based targets. In case of cancer, diabetes, you can choose only the human-based targets. If you want a druggable model, very specific and very experimental data, if you want, you can go for druggable models and pharmacophore models. So these last 2 would be in case of experimental data, if you need only those kind of data, then you can choose last 2 options. But generally, for cancer or diabetes, usually we select human protein targets only. And here you can see the 3,300. If you want to change the numbers, you can change the numbers, increase the number up to 1,000. So this is how we adjust the parameters and we check these parameters and submit option. So once you submit, you get the information like this. It means your data was submitted and you receive a mail for the submission option. Am I clear till now? Any queries about PharmMapper? Participants, any query? If there is no query, just give a thumbs up symbol. So let me proceed with the next topic. Hope there is no queries. So let me move with the next slide. So usually once we submit, we get the output. So I have given here for each column, what is the name, what is the name, parameter you get, and what is the different kind of score you get. And again, here also we have to find out the gene names for the targets. So that also I'll tell you when I show you practical. I have downloaded the Excel and kept the data. I'll just explain you the data definitely. Here you can have the pharma model, that is identifier for pharmacophore matched model, that is from the database, how many number of and how many pharmacophoric features were matched for the particular target with the particular with your input compound. And fit is nothing but you have higher the fit value, more more attached to the more the target and compound interaction. And normalized fit score, it was easy to compare across the model. And Z score value, more the Z score, more significant your match was. That is the target, whatever in the column was highly matchable. And number of hydrophobic, apart from that, all the pharmacophoric features, whatever we have mentioned, for that the values would be mentioned. Number of hydrophobic interaction point, number of hydrogen bond donor matched with the particular target, except that, and number of positive ionizable center matched with your target, number of negative ionizable center matched with the target, number of aromatic features matched with your particular compound-based target, and name of the protein. So this is the important thing we are going to retrieve. So this column alone we are going to take for our next study. And you have something called as class, that is the protein class, the enzyme class, or if it is a kinase-based protein, you would have the kinase. So based upon the name of the protein, we get in the name column. For that, the classification of the protein were given. And uniplot is nothing but uniplot ID, uniprot ID. Uniplot, it was mentioned as a column name, but it actually depicts your uniprot ID or the gene name. I'll just show you in the Excel, so it would be easy to understand. And biological role of the particular target. So if you want very much specifically, you want to fetch the information about the target. Suppose if I'm going to take top 20, 50 target, I need more detail about the particular target. In that case, I'll be checking this option and how it associated with this disease and therapeutic context. All these information will help me to get more idea about the disease and the functional information about the particular targets. Am I clear, participants? Any query? Any queries? So this is how your output of your PharmMapper would be. And the score value should be, Z score should be, that is fit would be raw alignment quality between your compound and pharmacophore model. And normalized score fit would be better for the comparison across the target. And the Z score, if it is greater than 1, it is better match. But if it is greater than 2, strong match with the target. So based upon your research you're going to carry it out, you can choose this threshold value and how much amount of target you get, and you can choose the target. So avoid taking the value below 1. So that would be better to check the scoring perspective. So these 3 criteria will help us to choose the better targets for our next level of study. That is high fit score, biological role, and this is linked. If you check all the data, then it would be better to take the targets. So with this pharmacophoric pharmapper database is over, I'll just show you again practically within the hands-on training. It's just a theory part. Any queries, participants, till now? Any queries, participants? Kindly unmute your mic and ask, or if there is no query, just give a
No,
thumbs
ma'am,
up.
no.
Yeah,
No, ma'am.
no queries. Okay, thank you so much. I'll be next. Okay, next database is Swiss target prediction. It is again based upon the structure feature. Again, it is another database to retrieve the targets for the compound. It is an online server. All the servers were the online, but here you get the target immediately. Whereas in PharmMapper, you will receive a mail maybe after 1 hour or 2 hours, or the next day you receive the mail based upon the server usage. So here it integrates your 2D fingerprint as 3D structure of your target to find out the targets. As we know, we have different kind of proteins. So for our compound, how much percentage of each kind of protein we are getting? Like this, we get it on the output page. And the key features, again, it gives a protein ID and chemical ID for the compound, and probability score would be there, where the nearer to 1 was the good. 0.8 above probability score would be better for your and research work if you take that data alone for your research work. Here it covers all kind of proteins, including enzymes, GPCR protein, ion channel, transporter, all the proteins, that is antigens, like all kind of protein data it covers. And here again, the output will have certain number of columns. So target information would be there, and common name, that is common name of the target, and Uniprot ID for the target, and ChemBL ID for the target class, and probability score, and the 2D and 3D structure, known actives. That is how many number of molecules able to bind with the target in the Swiss target. So that fingerprint data will also be available in addition to the probability score. So the probability score value would be 0 to 1. Higher the value, more the confidence, that is stronger the data set match would be. So this is how we interpret, that is, as I told, the probability score value. I have given the threshold value for selection of the targets. So greater than or equal to 0.7 would be better to take the targets with a particular range. If you have hundreds of targets, so for top 10 or top 20 targets with 0.7 above, you can take. So that would be better for our work. And in Swiss Target, again, this is the homepage of Swiss Target Prediction, where you need to paste your smiles format and just give once you paste the smiles format, here you have only 3 options. You can go and search for Homo sapiens, mus musculus, and Ratus novaticus. So defaultly, our work would be under Homo sapiens. So we go for Homo sapiens. And once you paste your smiles, you get the structure in the right-hand side. That is, in the drawing tool, you get the structure. Just give predict target, and you get the information in this perspective. So this is the information you are getting. The name, and the second column, common name, is nothing but your gene name. Next one is the Uniprot ID, ChemPL ID for this particular target. Enzyme class, whether it is a kinase enzyme, oxidoreductase enzyme. So based upon the enzyme class, you get the data. And probability value. So if it is fully green color, the value would be 1. If it is partially matched, the value, it would be half green color, or so that this will be vary with, if the probability score was very less, the green color would be very less. So, and last one would be the how many actives reported for 2D and 3D parameters. That was the last column. In this, but for this particular compound, it has 33% of oxidoreductases, kinases were reported, 20% oxidoreductase. So how many percentage of proteins were reported for this particular compound? So this is again quantitative. So it shows a good positive value. So for certain compound, for first target alone, you get complete green color. For remaining, you won't get the probability score value. So in that case, here very less targets only you get. Based upon the compound nature, again, your target value would be refined. So this small bio tools, once I complete with this, I'll just come back to this tool because we already took the disease-based targets and compound-based targets. Now we are going to identify. We are going to merge that. That is the last part. So once I complete with the hands-on session, I'll come back to this slide. Is that okay, participants? Shall we move on to hands-on training session? Any queries? Any queries in the presentation? If you have any queries, you can ask. No queries. Okay, thank you. So let me start with hands-on training session. Farm Mapper in Google, just type Farm Mapper. I'll just share you the link also. Those who are working, I'll just put the link in the chat box. So this page would be little bit slower. So once you give submit job, you get this page. And here, as I told earlier, we need a SDF file format. So I'm just clicking over the quercetin in the PubChem and just download the SDF file format. That would be the input to submit in this page. So as of now, if you are identified any compound, just try to submit the compound. Otherwise, just you also download the quercetin structure and submit in this database. So just go to download and just upload the structure and give your mail ID and give the compound name quercetin. So any queries in this page? So it is just browsing your data and just paste your email address and give your job description and give a continue symbol, continue. So it will go to the second page. So any queries you can discuss till now because this server would be generally little bit slow. Participants, let it load. So simultaneously, I'll just open the binding database. Is that okay? Because the server was very slow. I'll just tell you how to work tomorrow also. You can work slowly to get the data for Farm Mapper. I'll just show you the data. Can we submit? No, you have to submit each time single SDF file format, not more than one. And you can, maximum you can submit 10 job at the once. Once you get all the results, you have to go back from first again and you can submit the data. Okay. And coming to the binding database, so let it load because it was taking much time. So I don't want to waste the time. Simultaneously, I'll just open the binding database. here, as I told earlier, so in the specialized tool in the left hand side, you can see the specialized tool. And here you have find my compound targets. Okay, so once you find out that, I mean, if you click on to that, this page will be opened. So this is the parameters where you can see here the similarity value and IC50 value. So if you want here, usually we don't need to adjust any parameters. If you have SDF file format, you can browse and upload the SDF file format. Or if you have a Smiles format also, that is more than enough. So you can copy the Smiles format and paste it in the box over here. Click OK. You get the structured data over here. So once I get the structured data, here I am just going to adjust the threshold value to 0.7. Okay, and I'm just going to give go option. Hope you are also working participants. Second page open. For you, second page, if it is open, just check this PPT. It was very slow for me. I don't know why. You can see here, uh, select human target only and give submit option. That's enough. You no need to change anything. Just go to the human target only option and just give submit option. So if you want to change any parameter, just click on to this advanced option and check the parameters. And if nothing to modify, just click OK. Okay, done. You'll receive a mail in an hour, and I'll just show you the mail also. You will receive a mail like this once your job were done. And if you click on to this link, it will again open. For me, that is also very slow. If anybody can open this link, I'll just share the link, whatever I have received. So I'll just share it in the chat box so that for anybody, if it is opening, that would be fine because it was very slow to me. So you can see now it was actually actually I was trying for a long time. It was not opening, but now it was open. So you can see here the list of targets. So after the submission over here, you can see the output in the mail you will receive as a list of targets. Okay. So it would be around, so it was loading. So it will list out all the targets. So we'll have to wait for it. And so in the submission, as I told earlier, so kindly this option generate confirmer should be yes, maximum target. So you can increase or decrease the target number, generation confirmer. So select human target only. So I'm here, if you want advanced option, you just go and check. If you want energy minimization to be done, give yes, otherwise no. And here again, advanced option, hydrophobic donor acceptor parameters. If you want to adjust, you can go for this particular refining process. Otherwise, just give submit alone. Your data will be submitted and you will receive a mail notification receiving a mail like this. So once you receive a mail, you can open your data, and at the end of this page, you can download the Excel data. So it was loading. It was loaded till 1:38. So we need to wait for the data set. So as I told, you have the fit score, Z score value. So we have a mixed Z score value. So if you want to adjust the Z score value based upon Z score segregation, once the page were loaded, you can segregate the data based on Z score value, and you can take the top data or threshold data till 0.7 above you can take, and you can go for the analysis purpose. So I think I have got one query. For 100 compound, um, actually, it was a time-consuming process only. In case you have 100 compounds, you can go for virtual screening kind of thing. Come to a novel compound, then come to this approach. No, you cannot submit the, so, okay, in case if your which compound name you you chosen, Tanvi, because if uh, the compound doesn't have experimental data, it won't show the list. Acid. Maybe maybe for that particular compound, the data won't be available. So just now, just to find out the data, just check it for quercetin right now. Later, maybe you can work with your compound data, whether you are getting the data sets, we have to check. I also run simultaneously. For certain compound, the data won't be available, so that will also be a reason. So here you can see it was around 0.6 and 0.7 data. The targets were listed out. You can see top 60 targets were listed out here. So I have to take the each target. So suppose if I'm going to take, I need to prepare an Excel. So this I have to prepare it manually only. So you just try for quercetin right now. So that would be better. So here I need to prepare the file manually. I'll tell you one option. Suppose if I am going to copy the entire page data, I need to copy and put it in Excel. Getting a proper Excel kind of data. So manually, so there is no other download option available. So you have to do it manually for the target names. So here you get the target, just copy paste the targets alone in a separate column. After which you need to find out the gene name for each call, each target. That is, first column would be the target name. So the first column would be the target name, and next column would be the gene name. So suppose for this, I need to find out the gene name. I have to go to UniProt. So uniprot.org, it is again a database to find out the protein complete details. So once we analyze the target, finally we'll come here to get more detail about the target. So for this particular Homo sapiens, I got this gene name. So you can see the gene name over here, and you have to copy paste in the column. And next gene name, again nucleotidase, search Homo sapiens, NT5E, just copy paste the first gene name. So if you have multiple gene names, just go inside and check here. This should be your gene name. So if you have multiple gene names, because the same protein will have the different names, so there will be multiple gene names with the comma. So most probably take the first one. And otherwise, if you have more doubt in taking the gene name, so come here and copy the gene and paste and prepare a Excel-based data like this. Am I clear, participants? Again, the same results I tried for the components, but there it produced. Yeah, for a certain compound, maybe the experimental data not available. In that case, then don't go for binding database. Just submit in PharmMapper, and that is what I have given 2 to 3 database option, because this binding database is very much specific to the what to say, compound, target based, experimental based data. Hope I'm clear. Uh, so if you're okay with this binding database preparation, I'll be going with PharmMapper data in database interpretation. So you got your results for PharmMapper. That's good. Okay. So at the end of the page, you will be having the Excel download file option. Hope if you're able to download, you kindly download the Excel data. And I'll just show you. So this, I do have the result, but loading is here the loading was the issue. So I have the Excel file which is downloaded. So participants, if you're clear with binding database, let me move on to PharmMapper result explanation. Any queries with the binding database? For those who are working. Any queries? If there is no query, just give a thumbs up symbol so that I'll be going with next one. Okay, fine. So this PharmMapper database, once I downloaded the Excel, so it would be like a number kind of file, your job ID with the number, it will be downloaded. So this is the PharmMapper data for it. So first column would be pharma model. That would be a PDB ID for your data. And number of pharmacophoric feature matching here out of 5. How many pharmacophoric features were matching? That is based upon donor acceptor. How many pharmacophoric features were matching over here? Next column would be the fit, normal fit, and Z score value. So more the Z score, better your result would be. So avoid negative or a 0 below Z score. And this is your gene name. So this column is very much important, and this column. So these 2 columns are very much important. And so if you want to check more detail about function, disease, and indication, just check out this column. But as of now, so we are going to focus much on these 2 columns. Okay. First, so I'm not refining here with the score. I'm just taking the entire column right now. So here you have around 300 target. So 301 rows were there. So 1 row were not there. So 300 target. So as I have prepared over here, I need to prepare a data similarly. So this is from binding database. I have mentioned only 2 targets. So again, binding database. So how many target I'm getting from binding database? I have to take all. So this is from PharmMapper. So I'm just pasting all the targets I got over here. I'm just copying it and I'm just pasting all the targets. So here again, I need to prepare a gene names. So you can see Uniprot ID were there, or in the page of UniProt, you have this kind of thing. That is gene name followed by the organism name. So this data would be there. Similar way, here also gene name followed by your organism were there. So in that case, what I have to do, I have to remove these parts and I have to keep the gene name alone. Am I clear? So this column, I'm just copying it right now completely. I'll just tell you how to prepare a file slowly. Suppose if I'm going to, hope my screen is visible. Just going to prepare an Excel data. So for this prothrombin, THRB is the gene name. So I'm just deleting remaining part. For this, so I'm just preparing in this way for all the data. If this data is not here for this particular section, so in that case, what I have to do is I have to copy paste this ID in UniProt and search the data. So now you can see here the gene name. You have to copy paste the gene name and prepare. Am I clear, participants? So preparation part will get much time. So for each compound, if you're going to prepare a data, you have to prepare for the entire set like this. And the preparation part for the, even for a disease-based target, whatever it is, that will be time consuming here. So if you're going to refine with a score and come back, and again, you get some data set. Okay, I think here's some doubt, ma'am. When I pasted the smiles of stigmasterol on first page in binding database in search bar, I got some results. So your threshold parameter just adjusts to, if you definitely want some experimental structure for the data, so you can adjust threshold value till 0.4 and 0.5 above. I think you got some results. So that results, you just take the gene name and prepare the gene data sets. Whatever data you're getting, just go with the data set. Am I clear, participants? Till now, the preparation part, if I prepare entire data set, it will take time. So if you understand how to prepare, so let me proceed with Swiss target. Participants, any doubts? If there is any query, you can switch on your mic also and ask so that I can clarify. Hope there is no queries. So Swiss farm mapper target data prediction, is it clear till now? So like this, once you have done the preparation, just drag it. So you get the database name, protein name, and its specific gene name. So this data you have to prepare and you need to remove the duplicates, after which you need to merge it. So this part is very much important, participants, because this preparation part will take much time. So any queries you can ask right now. You can unmute and ask your queries. Shall I proceed with next Swiss target prediction? Is that clear till now? If yes, just give a thumbs up symbol so that I'll be proceeding with the next one. Anyhow, I'll be sharing the PPT so that you can work later also. Any query, participants, or is it okay? Shall I proceed with next tool? Just give a thumbs up symbol. Yeah, I got some. Okay, fine. Okay, so finally, I'm just moving on to Swiss target prediction. So again, here we have to paste the SMILES format of the compound. So as this particular compound, I'm just doing here. So just paste the compound, select Homo sapiens, just give predict targets. See to that, don't leave space after your pasting. It's only one small slide shows. You got a result for this 𝐏𝐥𝐸𝐴𝐶 acid. Yeah. So don't leave space after the SMILES format. So when you're copying the SMILES, be careful copying the till the oxygen atom or the data available data sets. Okay. So you can see here, it shows some green colors, variation in green color. So here you have again less number of targets. So you have around 100 entries, page showing 15 entries. So the column name would be your target protein name followed by your gene name and the probability score for the particular targets. Okay. So because again, if you're going to take lesser probability score, that is also fine because later we are going to refine our data with the help of function and all those things. But if you want very much specific data, then in that case, refine your data as much as possible. So I'm just opening this Excel file. Again, this database will list out the protein names followed by the gene name column. And you have the probability score value. Here it shows point. For few only it shows probability, others it shows 0. So you can take the values till whatever probability value was shown. So that value alone you can take, and you can take the gene names for that particular data sets and you can work with it. So I'm just copying these 2 columns for Swiss target and I'm just going to paste it here in the Excel I'm having. I'm sorry. So here you can see certain gene names, no issues. It has only one name, but certain gene names with the space, you have another gene name. So there would be FNTA, A chain, A and B, alpha and beta. Sometimes that would be, both the things would be there. So in that case, duplicate or you just find out the name for alpha and beta and insert a new column, new row and copy this and give it separately. So hope you understand what I'm telling. So if it has multiple name, just check whether it is an alternate name. So here you can see integrin alpha V and beta 3. So alpha V is first one. The second insert and you can copy this. This is alpha V, first one. And this is beta 3, second one. And copy the gene name. Am I clear what? And similarly, here also alpha and beta. So that difference were here. So this is how we try to find out the gene name from different databases. Any queries, participants, till now? So participants kindly unmute and ask if you have any queries. So as you asked for microorganism based, so in that case, you just choose in PharmMapper all organism. In binding databases, only human-based target would be available. But even if it is a human-based target, some targets would be, because even microorganism-based receptors, in human also, it will act on certain receptors. So when you choose pathways, there you can choose an option for human-based targets as well as microorganism. Also, in both the targets, you just check and you have an option to choose a pathway. In that, you can choose microorganism-based pathway in string database. There, I'll tell you how to refine pathways based upon disease or microorganism-based option. Is it clear, Shivan? But for microorganism, you won't get much targets as we get for human-based targeted research were going on still in that particular area. Is that clear? Okay. Any other query? So I'll just share a common file of target names. Suppose if I'm going to take this particular target, so as of now, I'm just taking few targets and just show you the merge option, because next class we are going to only work with the common genes that retrieve from already in the last class we have I have shown you how to retrieve a data for from therapeutic target database as well as from gene card, all those things I have shown you already. So I'll just show you that data sets. Just a minute. So this file I have already shared with you. So I'm just taking the liver cancer gene data set. I'll just copy this entire column. Okay. And I have shown you a Venn diagram kind of thing. So once I find out gene target for disease and compound data set. So I have taken data set quercetin and I have found out the gene names for it. And I have taken a data set of liver cancer and I have already told you to how to retrieve the data set and just paste the both the thing and click on to merge option. You get a Venn diagram like this. So in that you can see here a common gene name. That is this arrow mark indicates 46 number, right? So this is a common gene name. And this gene name will be listed in the right-hand side subset item listing. So there you can take and this Venn diagram. So for quercetin, if you get around 300 target and for liver cancer, you have around 4,000 targets. So for the common genes were around 46 unique genes, that alone you're going to analyze. Am I clear? So that data set alone we'll be analyzing and go for the next study. I'll just show you the Venn diagram. I'll just share you the link of this particular tool. So you can click onto this link and if you have gene data set, you can work. Otherwise, just listen. I'll just show you how to work. I'm just taking this particular column right now because as we so here you can see so this would be the home page for this Venn diagram. So you can use any comparator list tool for Venn diagram comparison. So I'm just pasting the column from Swiss target alone because as we have gene list over there. So I'm just pasting the gene name. So if there is multiple gene name, just click enter alone so that if it is on the same line, it will consider as the same gene. So just do this alone. And second column, as we have already predicted the gene data sets for disease. So I'm just pasting that. Just take the liver cancer alone, first title heading. And so here, just make it as liver cancer. And here, the compound name, quercetin or whatever compound I am just working, I have to type the name of the compound and click compare. So you can see here some 52 common targets for this particular, both the common genes. So I want to check this now. So I have to copy this and I have to go for string for the analysis. So I have found common genes by comparing two data sets retrieved from almost 6 to 7 databases and I got some data to analyze for the next level. So am I clear, participants? Any queries? Participants, any queries? Okay, no query, I think.
No, ma'am, no queries. Everything is clear.
Everything is clear. So you just try to prepare both the data sets as we worked almost 6 databases. Take the Saturday and Sunday for the preparation part, because as I told you, this gene preparation part and all, it will take time. I'll share a Monday, I'll share a example. I'll share an example file with you, format of the file. I'll just share a format of the file so that it would be helpful for you to prepare, so that you can list out from which database, and there might be common genes, same genes present in all the database. In PharmMapper, the same gene present in the Swiss target. So in that case, as I told you earlier, to remove the duplicate option. So you can entirely paste the column and remove the duplicate option and go for merging of the data. You get the common data set. For that data only, we are going to do the string data analysis on Monday. So Monday, we are going for a most important part, string data analysis. From there, we are going for pathway mapping. All those things will be done. But the initial preparation part, hope till now I'm clear about it. And any queries, participants? Any others? If you have any queries, let me clear it off. I'll share the file with you for your reference, and PPT also, so that you can work during the weekend. So in gene part preparation, any queries? Because that is the main thing, Excel preparation part. If you have any queries, participant can get cleared. You can switch on your mic and ask. If there is no query, we'll wind off the session. Participants, kindly fill your feedback forms that was posted in the chat box. Like anything, you can post your feedback regarding the session. So you can post your feedback in the link. If there is no queries, just fill off the feedback form. We'll wind up the session. I'll share you the PPT and the files relevant.